not connect to your lungs, it connects to your stomach. Trachea is what I meant to say. Okay, so where were we? Phagocytes. So here's a quick video that I want to show you guys of phagocytes in action.
Microorganisms that cause disease are called pathogens. When pathogens enter the body, white blood cells called phagocytes pass through the walls of the blood vessels and into the surrounding tissue. There they can ingest the pathogens and pathogens' toxins, absorbing and digesting them. Phagocytes can also destroy pathogens by releasing enzymes. Having absorbed a pathogen, the phagocytes may also send out chemical messages to call in other white blood cells.
Okay, so that's phagocytes. The final part of your innate immune response, so the non-specific immune response, is the inflammatory. Anytime your skin is ruptured or your body senses that there's some sort of infection, the cells at the site of that infection release signaling molecules. You know, we learned about cell signaling recently, same idea. They release these signaling molecules, and those signaling molecules go throughout the area and they keep dispersing. And whenever phagocytes come into contact with them, they go, oh my gosh, I should go over there because the signaling molecules are telling me I should go in that direction. They also increase blood flow to that area, trying to get those phagocytes to that area quicker, and that indirectly results in swelling, redness, warmth at that location, right? That's why if you get some sort of cut, like a really bad one, it might get swollen and red, right? Even if it's not getting like really terribly infected, but if it is getting really terribly infected, it's probably getting even more swollen and red. And so that's your inflammatory response to try to protect your body. Again, this is all non-specific. Okay, so we're going to actually skip toll-like receptors. That's not something you need to know for the purposes of our class. So moving on. So now we're looking at adaptive immunity. So this is the specific part of the immune system, the part where it's targeting one specific pathogen. So rather than just saying, oh, blanket, okay, it's self or not self, and anything that's not self, I'm going to keep out. That's the non-specific part of the immune system. This part is saying, okay, there is something that I know very specifically I need to attack. So for example, some specific virus, like the smallpox virus. All right, I know, like smallpox. I know specifically that I need to attack that specific thing. Okay, so that's what your body would be doing with the adaptive immunity. Okay, so adaptive immunity is related to B and T cells, as I mentioned before. These are also lymphocytes. So we have, again, here, image of lymphocytes. These ones are our phagocytes, and these ones are our specific. I was held of making my video for the third time. She's here. Okay, so I should keep filming. Okay, so anyway, so here are our white blood cells. So there's two kinds. There are our B cells and our T cells. Our B cells are related to our antibody response, and our T cells are related to the cell-mediated response. So the B cell part we refer to as humoral response, and that's just another, you can also think about it as the antibody response. T cells refer to the cell-mediated response. Oh, and one other thing I want to explain while we're on this slide is about what antigen means. So pathogens are, in this case, anything like a virus, bacteria, fungi, anything that's coming into your body that your body does not want there, that's being harmful to your body. Antigens are little they're little tags that say what that cell actually is, and that can be on a pathogen. It also could be on a perfectly good cell. But in the purposes of this lecture, we're talking usually about antigens from pathogens. We're talking about the specific immune response here. These antigen receptors are for a very specific antigen, and I'll explain what I mean more about that now. So this image, this figure, which is figure 35.16 in your textbook on page 723, is a very useful figure. It was also on the immune system worksheet that I passed out in class on the Friday before break. And it's going to be very helpful as you're trying to navigate sort of understanding this specific immune response, because there's a lot of different things and a lot. So it all begins with the helper T cell. The helper T cell gets activated because there's some antigens that an antigen presenting cell, which I'll talk more about those later. Basically, the cell is saying, hey, this is the antigen and of the pathogen that's attacking us. This is the little name tag. You would think of the antigen as sort of like a name tag. Here's the name tag of the pathogen that's attacking us. You need to seek out this pathogen. Here's its name tag. And the helper T cell goes, okay, I know I've been alerted, so now it's activated, and it then in turn activates B cells and cytotoxic T cells. So we'll talk about what those are now. So moving on, we're going to first talk about B cells, which is to do with the humoral response. So I've drawn, this is what I would draw if I was had a whiteboard, right, and this was in class. So this is something I'm going to keep referring to, and I would encourage you to have a diagram kind of like this in your notes, right? So here we are with our helper T cells, and we're going to activate our B cells for the humoral response, or the antibody response. Okay. So the humoral response, antibodies are proteins that are specific to one specific antigen. So they have a part of them that binds only, like puzzle piece style, only with one specific antigen. Okay, so here's sort of the sequence of events. We have a helper T cell here. We have a phagocyte here. The phagocyte has ingested a pathogen. Here we see that happening. The pathogen is broken down. There are little antigens that are taken off of it, that little name tag, and the phagocyte holds out the name tag and says, hey, helper T cell, you should go and make things to attack this specific cell because it's a bad guy. And then the activated helper T cell says, B cells, you should start making some antibodies. And so then the B cells, memory B cells, and the plasma cells make antibodies, and they go off and do their job. The memory B cells, we'll talk about more in a second. So here we have basically what I just laid out. Plasma cells, they make antibodies. That's their whole job. Memory cells, basically they stick around in case you have a future infection, this idea here. So if we look at my little diagram that I would have on the whiteboard, right? So here we have our B cells. This is the humoral or antibody response, and we have our plasma cells, and we have our memory B cells. So plasma B cells, and memory B cells. Plasma B cells make antibodies. Memory B cells stick around in case there's another infection of the same thing, right? Not just any other infection. This is not non-specific. This is very specific. Another infection of the same thing. Okay, so antibodies. How do they work? So you don't need to know about this heavy chain, light chain, constant. You don't need to know about any of that stuff. But what you do need to know is antibody is specific to a specific antigen. So if we look here, right, there's this antigen binding site, and that's going to fit like a puzzle piece. So the antibody, this is not just, oh, whatever antibody I have can work for whatever problem I have. The antibody is binding with a very specific pathogen. So basically how this works is your body is able to produce, basically because of genes, and we haven't talked about sort of this aspect of exons and introns and all of that, the DNA stuff we haven't talked about yet, but basically each of your genes for B cells, inside the B cells for making the antibodies, which are a protein, each of those genes can produce 100 different millions of different variations of antibodies. And they just produce all these different types, constantly producing all these different types of antibodies. And then, and then they have clonal selection and they produce a whole ton of it. So that's what this is explained in this image. So we have a whole bunch of different types of antibodies. All these different types of antibodies are produced in the B cell. which one fits, and this one fits. So then we have clonal selection, and they make a whole bunch of those B cells. So that specific B cell, since it's producing that specific antibody, gets produced over and over and over. And it gets produced as plasma B cells and as memory B cells. The memory B cells stick around in case of future infection, and the plasma B cells produce a ton of antibodies. They're just like antibody producing factories. Produce tons of antibodies. So here's this video, which is pretty interesting, but I'm not going to have us watch it during the lecture, right? Because like you can watch it on your own. So you can click on the Prezi that I put a link to in this post, and you can watch this video. It's about sort of a visual for clonal selection, and it's pretty cool. Okay, so moving on, once the B cell has an antigen receptor that's specific to the antibodies, and then what's the about how all these antibodies get produced, but why do we care? So this is what antibodies do. They have three effects that they can have on a pathogen. The first is opsonization. Opsonization is the pathogen being tagged by the antibody for phagocytes. So basically, macrophage, by the way, is like a big phagocyte. So here we have our pathogen. Here we have our antibody. Here, the orange thing, that's the antigen. So the antibodies are tagging macrophage, come on over here and eat this guy. It's not good. Then the macrophage comes on over and is like, ooh, I see all those antibodies. That must tell me I should eat that. And then they eat it, right? So that's one option. Another thing antibodies can do is neutralization, which is basically, so here we have our virus. This is our pathogen. Orange, that's the antigen. Purple, that's the antibody. So neutralization is basically the antibody goes all around whatever the pathogen is and makes it so the pathogen can't actually do anything. Now the virus can't infect any cells because it's completely surrounded by antibodies, so it's neutralized. And then the final option is complement activation, which is where you have your antibody, it binds to the antigen on the pathogen, and then something else in turn binds to the antigen, and that something else actually triggers lysis. It makes that cell eventually explode or kill itself or die. Okay, so that's our antibodies can work. Our last thing about the humoral response is this here. So this image is actually super helpful. I would definitely recommend it. Is that in the textbook? I'm sure it is, but actually I didn't flag the page. Oh, yes. It's on page 720, and it's figure 35.12. So this figure has to do with your primary and secondary response, which has to do with your memory B cells. So, oh no, I don't want to go that way. I want to go back here. So, basically, you're first exposed to this pathogen. And pathogen has antigens, and so your antibodies are formed to bind to the antigens, and you slowly figure out, okay, these are the antibodies that we need. We're taking some time to find the right one, and then we find it, and then we make a whole bunch of them, and now eventually we get that pathogen, and yay, we're going back to healthy. We don't need to make the antibodies anymore. Great. Okay, this pathogen A. You also get exposed to pathogen B, an antigen B. So for antibodies for pathogen A, they spike. You produce a whole ton of them. Okay, and that means that actually you're not even going to feel sick because you produce them so fast. You don't even feel that. You don't feel any sort of produce so many antibodies. You nipped it in the bud. But for pathogen B, you're definitely going to feel sick because you have to go through the whole process again. So this is why memory B cells are so important, because we have memory B cells, that's why we're able to produce so many antibodies right away for that specific pathogen, right? So again, if we look, we've done all of this side here, the humoral response. Okay, so next we'll do the cell-mediated response.